Hello, and a very warm welcome to today's Nucleic Acid Insights webinar titled Implementation of an Improved Platform for Downstream Processing of Plasmid DNA. I'm Kate Fabricius, and I'll be moderating today's webinar. And joining me are Justina Martinkiene and Klemen Božič, who will provide insights into optimizing a process for the capture of pDNA and showcasing data on a novel approach to remove undesired pDNA isoforms. After the presentation, we'll have a live Q&A session, so feel free to pose your questions to our speakers using the Ask a Question box at the bottom of your screen, and we'll try to get to them during the session. I'd also like to draw your attention to the resources on the right-hand side of the screen, where you can find more information on the topics covered today. Now I'd like to introduce our presenters. Justina is an associate scientist at AstraZeneca in the Purification Process Sciences team, where she is involved in process development activities of various modalities, pDNA, antibodies, and mRNA. Klemen has been a process development scientist at Sartorius BIA separations for 4 years and has extensive experience in pDNA analytics and pDNA downstream process development. So without further ado, I'll hand over to Justina to kick off today's presentation.
Hello, everyone. Thanks, Kate, for the introduction. So today I would like to talk about how we have enabled symmetrous DNA codons at scale for plasmid DNA capture. So, in today's agenda, I will give a background of pDNA downstream processing and analytics. Then I will talk about our plasmid DNA purification process 1 that we have initially developed and used for plasmid DNA purification. And also I will provide what the drawbacks this process has and why we wanted a new process to be developed. Then I will talk about the new pDNA process that we named version 2 development, where we have enabled monolith columns. And then I will provide some comparison between those two processes and give some conclusions. So just a little bit of a explanation about plasmid DNA journey in the biopharmaceuticals department that I am representing today. In our department, we use plasmid DNA in the variety of scientific areas and fields. Plasmid DNA journey in our department started in about 2018, where we started focusing on so-called in vivo expressed biologics, such as plasmid DNA, where pDNA can be one of the platforms to express IgG genes. Later on, especially after the COVID pandemic hit us, company strategy have changed a little bit. We started focusing more on the mRNA, and we started purifying our plasmid DNA that would later go through the enzymatic digestion, where we would generate linear DNA isoform and use that isoform for the mRNA production through the in vivo through the ADT reaction. Speaking about the supercoiled, speaking about the pDNA in general, it's important to highlight that even though pDNA naturally in the cell exists in a variety of different isoforms, we are mainly focused on the supercoiled pDNA isoform. And we want in our pDNA samples this form to be expressed in more than 80% purity. Also, based on the FDA guidance, we have specific requirements for the variety of impurities. So we want to design our process that would meet those criteria for the quality of supercoiled and for the levels of impurities. Additionally, what we need to pay attention to is that we want our process to be scalable. We want our process to be able to handle a variety of different size of plasmids. Additionally, as a downstream process team, we want to have solutions for in-process sample filtration. So, in order to develop this type of process, we have to have a plasmid DNA quality and purity monitoring tools. And for that, we have mainly three analytical tools enabled in AstraZeneca. We use agarose gels. Well, this technique is, I would say, quite aged. So there are occasions where we use agarose gels. There are some projects where we still run them, but mainly what we do is anion exchange HPLC tool, where we have enabled Cmax pDNA monomer columns. This is our routine testing that we do on our samples. And then another tool is the capillary gel electrophoresis, CGE. Typically, this test is done by our analytical team, and this assay is our lot release technique method. What else we do is sometimes, if we see that we are dealing with starting material, E. coli material, ISIC material, that contains different isoforms that we don't know about, or we are not sure which peak represents which isoform. What we sometimes also do, we do enzymatic digestions with the enzymes. We generate isoforms ourselves, and then we can do comparison and identify those isoforms in our samples. So now let's move to the plasmid DNA purification process that we have developed initially and used for the variety of constructs. In this slide, you can see the whole process flow, and also there is small explanation what each step does and is responsible for. So this process, version 1 process, was successfully used to purify a range of high-quality plasmids at tox scale with fit-for-purpose process controls as an intermediate product. This process delivers recoveries ranging from 30 to 60%, and the recoveries ranges from 3 to 10 grams of pDNA from 100-liter reactor. Unfortunately, even though we've used this process quite successfully for our constructs, it has some bottlenecks. And one of the main bottlenecks is, unfortunately, low binding capacity on the capture step. And that's the reason why we sometimes have to perform large cycle numbers. So it requires us multiple manufacturing slots in order to proceed the full 100-liter bioreactor. Additionally, what we noticed while working with a variety of different plasmid DNA constructs, that we are getting more and more complicated constructs. The sizes are getting larger. We are seeing increasing levels of impurities, as well as we sometimes see concateners in our starting lysic materials. All of those reasons asks us to develop a new pDNA capture step and a new pDNA purification process. So that's why we started developing a pDNA purification process version 2, where we started implementing monolith columns. So first of all, in order for us to develop a new purification process for the pDNA purification, we obviously didn't know that we will end up using and implementing monolith columns. what we did was we looked at the what chromatography media vendors are suggesting to use, what they have developed for the plasmid DNA capture. We kind of made a table where we put all the available information in one place, and we started doing evaluation and screening studies of those resins and those media and membranes. So in this slide, you can see comparison results between the media that we have been evaluating. So let's start from what we were using in those studies. So first of all, in all of these ones, we were using same construct, pDNA construct, that the size was, I think, approximately 6.9 kb. Then what we did, in order to elute pDNA that did bind to those media, we were using sodium chloride gradient ranging from 0 to 2 molar. And in the first round, that is highlighted in purple color, you can see data of the yields and purities where we did load crude E. coli lysate. So it means we would just lyse the cells. We didn't do any lysate adjustments, and we would just load on the media. And as you can see, if we look at the yields, purple, that are highlighted in purple, just monolith showed very high yield. And for the other media, the yields were not so impressive. And then if we look at the purity, which is in the dark blue color, purity unfortunately was not so great even for the monoliths. And then we did another another study, and another set of experiments. Those are highlighted in yellow color. And in those experiments, we did conditioning of the E. coli lysate, where we used sodium chloride, and that significantly improved the binding and improved our yields for almost each media that was tested in the study. And the purity improved quite a lot, especially for the monolith columns. And that's why, as you can see, monoliths, 6 micrometer channel monoliths have demonstrated best performance, and that's why we did choose monolith columns for the further development. So, what this experiment that we just discussed showed us, that we need to do some sort of lysate adjustment that would help us with the binding and eluting our pDNA. And after discussions, after the literature review, we have decided that instead of sodium chloride, we will use calcium chloride. That really helps and improves our pDNA quality. In this slide, you can see that sodium chloride, unfortunately, is not removing impurities, but calcium chloride, you can see, did a huge, massive improvement in the quality of pDNA. And so after we decided that we will be going with the calcium chloride for the precipitation in coffee purities, we then made a plan that we will be developing first E. coli conditioning with calcium chloride strategy. Then we need to remove calcium chloride from our lysates, and for that we did choose lysate UFDF step, and we were developing this step. Then we have to confirm that the newly developed ion exchange step on simultaneous DA column works across different sizes of pDNA constructs. And we have to confirm that the process, new process version 2, is scalable. So in this slide, you can see that we looked at different calcium chloride addition strategies. We did test a range of calcium chloride concentrations ranging from 500 to 100 millimolar. We also looked at pDNA stability after we add calcium chloride. That is very important for us. And finally, we did agree that the third strategy will be final. So what we do now in our process, we would do alkaline lysis. We add calcium chloride, we perform clarification, then calcium chloride removal via TFF step, and then we load on the monolith column. Then for the calcium chloride removal, as I said, we've chosen to do UFTF step. And for the optimization and development of lysate UFTF step, we did use a system from Sartorius called Amber cross-flow system. This system allowed us to look at different membranes, different membrane chemistries, different cutoffs. We found that 300 kilodalton membranes leads to substantial clearance of RNA and RNA impurity, which is majoring in our lysates. And we have successfully transferred our developed TFF conditions from 10 centimeters square membrane from amber to the cross-flow system on Helicon 88 centimeter square membranes. So now, once we developed our calcium chloride addition strategy, our calcium chloride removal strategy, we wanted to confirm that the developed anion exchange step on monoliths works across different constructs and is scalable. So we first did initial experiments with two different plasmid DNAs, 4 and 12 kb plasmids, utilizing 1 mil and 8 mil columns, and then we successfully have scaled up the process to 18 mil column. And in this slide, you can see the graph that represents recoveries of anion exchange step for a variety of constructs, more than 10 pDNA constructs, where we get always very good recovery, more than 80%. And then we successfully transferred our process to even higher scale, to 800 mil scale. And in this slide, you can see comparison from the data from 18 mil one and 800 mil one with two different constructs, where the recovery was respectively approximately 89% for 13.6 kb construct, and we got about 91% supercoiled, and for the 6.4 construct, we got 100% recovery on 800 mil monolith column, and the levels of impurities were meeting criteria from the FDA. So now, just a quick comparison between those two processes, version 1 and version 2. You can see both processes in the slides, what steps we do that we just discussed. So we significantly improved the plasmid DNA capture step capacity, which was previously a big bottleneck for us. So we achieved 5-fold increase. We increased our plasmid DNA productivity quite a lot. Plasmid DNA capture step recovery also increased, as well as overall process recovery. And process duration in days, the version 2 process, unfortunately, is a little bit longer. So just to conclude, so in this resin membrane study that I just described, simultaneous DNA columns have demonstrated best performance. That's why those columns were chosen for the further development. Simultaneous DNA columns were successfully implemented and scaled up for plasmid DNA capture step. In pDNA purification process 2, delivering required pDNA purity levels and impurity clearance across different plasmid DNA constructs that are ranging in the size from 3 to 13 kb. We got 5-fold increase in pDNA binding capacity compared with the process 1. And that increasement allows us to reduce batch processing times and our buffer consumption, so environmentally friendly change. And average pDNA capture step recovery increased from 72% in the process that we have in process 1 to 100% that we have now in process 2. That's all I have. I think I just give a quick massive thank you to my colleagues in AstraZeneca that did a massive job here, trying to implement monolith columns, do all those experiments, and also a big thank you to bioseparations team for the valuable consultations and support on this work.
Okay. Thank you for the word. I'm going to talk about selective hydrophobic interaction chromatography for high purity of supercoiled DNA plasmids. I'm Klemen Božič from Sartorius Bioseparations, located in Slovenia. Excuse me. So we are producers of SIM monoliths and Process Development Center for Advanced Therapies. I'm a scientist in plasmid and mRNA process development team. Our facility is located in a small city, Justina, right in the center of beautiful Vipava Valley, surrounded by mountains, sunny vineyards, and many natural attractions like caves, water springs. So you're all warmly invited to visit us. But let's go first through our standard plasmid downstream process workflow. So similarly to what Justina described before, alkaline lysis of resuspended cells is followed by calcium chloride treatment and clarification. Clarified lysate may then be prepared for DAE capture via TFF or simple by dilution with water. But the plasmid eluted from DAE is then polished by hydrophobic interaction chromatography as such, so when we need supercoiled plasmid at the end, or after linearization in case of plasmid template for mRNA production. The entire plasmid downstream process is monitored by at-line analytics using our in-house pDNA pDNA platform, which is a powerful analytical tool comprising analytical columns and standards, customized system, and methods. So all together in a user-friendly format. Let's now focus on polishing of the supercoiled plasmid. There are two aspects of polishing procedure that have to be considered. First one is the quality of supercoiled plasmid. Polishing step has to increase the purity of supercoiled plasmid by removal of impurities. So here are listed by increasing hydrophobicity. So it would go from open circle plasmid, linear plasmid, and genomic DNA. Then we have host cell RNA, endotoxins, and host cell proteins. And somewhere in the middle, we have also our target molecule, supercoiled. So its hydrophobicity is slightly higher than the one of linear species, and that's why it's very tough to discriminate between them and to separate them chromatographically. Host cell RNA, endotoxins, and host cell proteins are pretty more hydrophobic than supercoiled, and that means that they actually remain on the column during the elution phase of supercoiled. The second aspect is more process related. So we need to take into account that it's highly appreciated. The process is generic to plasmid size, to the amount of material loaded, to the load of impurities. It has to be simple to scale up, environmentally acceptable, and last but not least, it has to be GMP compatible. So on this slide, let's say this is our starting position. I'd like to show how open circular and supercoiled isoforms of plasmid are separated in descending linear gradient of ammonium sulfate on CMC4HLD column in case of standard bind dilute approach. So we load in high salt, everything, so our target molecule with impurities, and then we try to separate them in descending ammonium sulfate gradient. We see that resolution between open circular and supercoiled is good, even at high flow rate, like 5 column volumes per minute. But there are also several other benefits of using SIM monoliths. For example, high capacity, very, very low shear stress in comparison to other supports, which is especially important with large biomolecules like nucleic acids. There is also a possibility to reuse them. That's why they are very, very appropriate for some process intensification approaches like cycling, etc. So these are all the benefits why monoliths are very, very beneficial for working with biomolecules. So there are at least 3 approaches or strategies of using CMC4HLD column for polishing of plasmid. Classic one, already described before, bind dilute. We loaded high salt, everything, impurities, target molecule, and then we can partially separate our target molecule from impurities by applying lower concentration of ammonium sulfate. It can be either in linear or step elution. Unfortunately, approach is not able to completely remove open circular, so there is some coelution of open circular and also of linear plasmid and genomic DNA. We see at the starting or at the beginning that linear species are very, very similar to the target molecule regarding the hydrophobicity. Second approach is sample displacement. Here, lower salt concentrations, so below 2 molar ammonium sulfate, are used, and supercoiled plasmid is enriched over open circular by displacing it when column capacity is exceeded. This can be achieved because supercoiled is more hydrophobic than open circular, but only at certain and very finely tuned conditions, which is not very convenient for production processes, scale up, and so on. Today, we would like to present a new approach, which is selective hydrophobic interaction chromatography. Here we use even lower concentrations of ammonium sulfate, preventing the impurities, so open circular, linear plasmid, and genomic DNA, to bind to the support. So we actually separate them from target molecule already during the loading phase. We do not even bind them to the column. Importantly, approach is generic to amount of load, so it doesn't matter if you underload or overload the column, the eluted plasmid will be of the same purity. It's generic to load of impurities, so it does not matter how much unwanted species there are, and it's easily scalable. Let's see some examples of its use. So first of all, we want to remove open circular plasmid, which is the less hydrophobic and, let's say, the most simple to remove. First example is was 7.5 kilobarrels plasmid with 33% of load of open circular in load. We tested several different conditions, how to improve the purity, and we used concentrations of ammonium sulfate between 1.55 and 1.85. Sample was prepared by addition of stock solution of ammonium sulfate to increase the ammonium sulfate concentration. Additionally, in one case at 1.7 molar, we prepared another sample where we added another part of loading buffer. So we actually reduced the concentrations of sodium chloride, which originates from the previous chromatographic steps, so from the anion exchanger. Elution from anion exchanger includes some sodium chloride, which represent a secondary salt in our system. It can also contribute to the cosmotropic effect of salt, which promote then binding to the HIC column. we see that we overloaded the column. We have 2 breakthroughs, and CMAC pDNA chromatograms and agarose gel show that we removed impurity in the flow-through and eluted pure supercoiled plasmid. So the aim was achieved, but as we want to say that the approach is generic to plasmid size, we would like to show also that it works with larger plasmid, which was 11.6 kilobase plasmid with 19% of open circulating load. We used similar range of ammonium sulfate concentration, but this time we prepared all samples with additional dilution. So we reduced the concentration of sodium chloride from the previous step. And we see that actually all of tested conditions resulted in really enriched supercoiled, but 2 were, let's say, outstanding. The lowest 2, so 1.55 and 1.7, which is actually corresponding to the previous slide with 7.5 kilobase per plasmid. We can say that similar conditions resulted in similar purity of our eluted plasmid. So approach is generic to plasmid size. But especially when scaling up the plasmid downstream process, some steps become challenging. For example, batch alkaline lysis, poor control over lysis conditions, for example, contact time with sodium hydroxide, mixing, and so on, result in increased amounts of host cell impurities, including genomic DNA. And HIC is actually a perfect tool to get rid of it. As an example, we have 6.7 kilobase per plasmid, which contained at the beginning approximately 1.8% of genomic, confirmed by qPCR, and let's say 2% of open circular linear plasmid in load. This time, we reduced the concentration of ammonium sulfate in our experiments because we know that linear species are more hydrophobic than open circular, so we need to get lower with ammonium sulfate to put them into the flow-through. And we see here that practically all tested conditions from 1.25 up to 1.55 molar resulted in practically complete elimination of genomic DNA, and at the same time we see that the CMAC PATFIX chromatogram show us that also other plasmid isoforms were removed. We have only pure supercoiled in L8. There are also other impurities which need to be removed. So besides genomic DNA, we also tested removal of endotoxins and host cell proteins. In-process samples from several preparative runs from different scale, from 1 to 400 ml column were analyzed. But to simplify the presentation, we only show data for 2 sizes of plasmid. And I think that it's clear that we can confirm that all impurities, including genomic, endotoxins, and host cell proteins, were reduced to below LOQ or to below acceptable limits. What about process properties? We say that HIC is scalable, and we want to prove it. And the first example is a test on a, let's say, lab scale column, 40 ml, with 4.7 kilobase per plasmid, with 23% of open circulating load. Also here, we exceeded the column capacity, and which resulted in complete removal of open circular during the loading phase and pure supercoiled in our elution. Dynamic binding capacity of this experiment was determined to be 2 mg per milliliter of monolith. We need to highlight that in this approach, in this mode, all the column capacity is practically used only for your target molecule. So let's say it's not misused with impurities which would bind to the support in case of higher salt, higher ammonium sulfate used. And this means that actually the benefit of this approach is even higher in case of more impurities, because you avoid binding them, so you can really use all the capacity only for your target molecule. But we went further to a pilot scale, to 400 ml column with a bit larger plasmid, which was 7.3 kilobase per. It had some open circular and also some linear isoform in load. This time we only loaded up to approximately 50% of column capacity, so there was no second breakthrough, and still we can see from the pad-fix chromatograms that eluate is pure supercoiled, while open circular and linear species were removed during the loading phase. So elimination of impurities was successful, and our approach can be declared as scalable. Another important benefit of this approach is that it reduces ammonium sulfate consumption. Using HIC instead of standard bind dilute approach reduces consumption of ammonium sulfate by 50%. If we prepare the sample for HIC with TFF, so that we concentrate the eluate from anion exchanger and exchange the buffer, we can reduce the ammonium sulfate consumption by approximately 75%, which significantly reduces disposal, logistic challenges, and cost of goods. Moreover, by using this approach, we can avoid loss of plasmid on pre-column filtration steps. So we have much lower concentrations of ammonium sulfate, so less hydrophobic conditions, and this way we can prevent loss of plasmid, even for large size plasmid on process filters. So let's make some conclusions at this point. We started with the challenge, actually several challenges, which are removal of impurities like unwanted plasmid isoforms, genomic DNA from E. coli, and other host cell impurities. And last but not least, also high consumption of ammonium sulfate represent a real challenge on a production scale for all the customers. But we found a solution, which is highly selective for supercoiled plasmid. So for the target molecule, it's generic to plasmid size, to load of impurities, and it represents a robust process with good scalability options. By this approach, you also get HIC results, which are more than 95% of supercoiled plasmid, and at the same time, impurities are reduced below limit of quantification. We can reduce the consumption of ammonium sulfate per gram of supercoiled plasmid by more than 50%, and we also avoid loss of plasmid in filtration steps. And so connected also with Justina's presentation, we can here say that Sartorius bioseparations offers complete solution for purification of plasmid. It's called HIP2 plasmid process pack and includes not only columns, but also protocols which are developed in-house here in our process development team and then transferred to the customer site. This process package is agnostic to upstream productivity and feed material quality. It can be used for production of supercoiled plasmid, which is then dedicated for transfection or for vaccines, or linear plasmid in case of IVT templates. We ensure the removal of contaminants with 2 chromatography steps, which definitely improves patient safety. It is completely scalable from mg scale per cycle to more than 200 grams per cycle. And on the other side, it also offers the possibility of some high throughput screening units like 24 or 96 well plates. And at this point, I would like to thank you all for the attention. And now I'm happy, or we are happy, to take some questions also from the audience. Thank you.
Thank you, Klemen and Justina, for a great presentation. Now I'll ask you to join me on camera for the Q&A. Klemen, do you just want to stop sharing for us, please?
Yes.
Perfect. I was going to say, Justina, can you stop sharing as well, please?
Yes.
Great, fantastic. So the first question is for you, Justina. What sodium chloride concentrations did you use to condition your lysate?
I assume this question was from the studies that we did where we were analyzing different chromatography media. So actually, we are targeting not the specific sodium chloride concentration, but we are targeting specific conductivity that we are achieving by doing the spiking of the lysate with sodium chloride. And that conductivity we have developed through a lot of experiments, and unfortunately I cannot tell exact numbers here.
No, that's totally understandable. Thank you for that. The next question, also for you, Justina. What other process development activities are you planning to optimize and further improve the version 2 pDNA purification process?
Yeah, so we know that our E. coli lysis process is not perfect. So in the future, we might look into this step and improve it a little bit. We know that Sartorius already have developed a equipment called alkalizator that is really nicely helps with the lysis, with the addition of calcium chloride. We might look into that in the future. Not sure yet if we will, but another step what we could look at is our second chromatography step, the polishing step. It's also not ideal, so we might look for alternative resins. We already did some screening studies. We have some chromatography tools in mind what we could try.
Perfect. Klemen, the next question is for you. In what case is this approach the most beneficial in comparison to standard bind-elute approach? Klemen, I think you might be on mute. I can't hear you.
Excuse me. Thank you for the question. Yeah, as mentioned already during the presentation, so we know that if we have more impurities, especially from large-scale processes, for example, genomic DNA and others, that would occupy part of your binding capacity in case of bind-elute. So, okay, overall capacity may be higher because you use higher concentration of ammonium sulfate, but at the end, only part of it will be used for the target molecule. So, especially in this case, this novel approach is beneficial because it lets the impurities to go into the flow-through, and that way they do not occupy the binding sites on the column. So all the material, all the capacity is only used for the target molecule, and not only this, but you actually improve the quality, the purity of the supercoiled plasmid at the same time.
Perfect, thank you. Justina, another one for you. How long did it take for the team to develop a new purification process version 2?
So I would say it was about 1 year, 2 months. So for the selected media evaluation experiment, we took approximately 2 months, and then the whole simultaneous DA column development, process development, including the calcium chloride addition strategy development, took us about 8 months, and then I would say 4 months took for us to transfer the process to our scale-up people and successfully run on the 800 ml column, because in our sites we do runs up to 80 ml size of monoliths.
Thank you for that. Klemen, for you, the next question. Is the HIC approach applicable to different types and sizes of plasmids?
Yes. Besides the examples I showed here, you can find more of them in the article which we published on this topic. So we tested plasmid size from 3 to 12 kilobase pairs. These plasmids were some of them intended for further linearization, so there were actually mRNA templates or plasmid templates for mRNA. Some of them were then used for transfection, and so depends completely. But HIC is a very, very robust and let's say generic approach, and it does not matter what kind of plasmid you have to polish.
Thank you. That's really interesting. Another one for you, Klemen. Does the approach remove gDNA?
Yes. As explained during the presentation, gDNA, so it may be, of course, of several sizes here, because the lysis conditions damage it, and you can get different lengths of this. But in principle, it's a linear DNA, and in that aspect, it's similar to the linear plasmid, and it has also pretty much similar properties. And it is tough to get rid of it, so that's why bind-elute approach is not so efficient here, because the genomic DNA co-elute with your target molecule. But if you do not bind it from the beginning, so that you do not bind to the column, which we do with HIC approach, then you can get rid of it. Yes.
Okay, that's good to know. Justina, why do you think other tested media, for example, chromatography resins, membranes, were demonstrating lower performance, especially in terms of delivering lower purity?
So in this whole screening of the media resins membranes experiment, we, as I mentioned in the slides, we were looking in two conditions, where the lysate was crude and where the lysate was conditioned with sodium chloride. So where the lysate was crude, obviously, I think it's just for those media and resins, it was challenging to deal with all kinds of impurities that we are loading on the column. And then where we increased slightly the performance with the conditioned lysate, unfortunately, we've used just one conductivity that I've mentioned previously, that we are targeting not the specific concentration, but specific conductivity. And that conductivity value was developed for our initial capture resin that we are using in our version 1. It's not designed for those other tested media or resins. So it simply might be that more work, more development work is needed to find best lysate loading conditions for those resins or media to get similar results that we saw for the monolith column.
Thank you. That makes a lot of sense. Klemen, for you, how much pDNA can be processed in a single cycle?
Yeah, depending on the column size you use, I would say. But if we presented before that capacity, we were up to 2 mix per mil. So if you use 8-liter column, that would mean that you can, with one cycle, you can polish 16 grams. Would that be the answer? This is actually not the largest, but 8-liter is actually something which is a catalog product. But yeah, there are also 40 liters columns, but which are custom made on request.
Good. All right. Is this GMP compatible?
Yes. The approach is completely GMP compatible, so it has no limitations for implementation in GMP environment.
Thank you. And one other one for you here, Klemen. How do you select concentration of AS for loading in HIC conditions?
Yeah, thanks for these questions. It's so depending a bit on what is the quality of your starting material. So you need to have a good insight in the quality of the material you're gonna load. If you can confirm that there is no genomic DNA present in your starting material, then you can afford to use slightly higher ammonium sulfate concentration, and that way you will actually not remove linear species, but if there are none, it's not a problem. And so you can still enrich your supercoiled over open circular. In case that your product includes also linear species, then you need to go lower. Otherwise, you will not be able to remove this unwanted species from your sample.
Thank you for that. This audience member would like to know what are typically sources of your pDNA, which cell types, for example. I'm not sure who they were directing it to, so I don't know which one of you would like to answer.
From my side, the answer would be that it's E. coli, DH5 alpha, at least majority of materials we work with.
Yes, so from my side, yes, E. coli is the same as cell line, yeah.
Perfect, thank you very much both. This audience member has sent in a question. We have problems about AEX binding capacity and clarification. What did you try the way for clarification step before chromatography step? Who would like to take this one?
So if from my side, as I've mentioned, calcium chloride precipitation for all kinds of impurities was really helpful for us. Klemen, do you have anything else to add on this?
Yeah, but regarding binding capacity, so it's similar as with polishing steps. So the more impurities you load to the column, the lower will be capacity for your target molecule. I'm talking here about host cell RNA. So if it's not removed or precipitated during the lysis step, then you'll introduce it to the anion exchanger. and it will use part of the capacity. So if there is high amount of RNA, capacity for plasmid will be lower, especially with plasmids which have low copy numbers of plasmid, for example. So the ratio between RNA impurities and your target molecule is very, very high. It's even more pronounced. So yeah, actually the removal of RNA, I find it is crucial already during the lysis and clarification.
Perfect. Well, thank you both for answering that question. Thank you, Justina and Klemen, for answering all those questions. That's all we've got time for today. Any questions we didn't get to, we will reply via email. The webinar will be available on demand tomorrow, so look out for an email from us with the link. We'd also like to invite you to take a short survey to provide us with feedback on today's webinar, the link to which can be found in the resources on the right-hand side of your screen or in the on-demand email that we'll be sending out tomorrow. All that's left is to thank our speakers once more for great presentations and to thank you, the audience, for listening. We hope you'll join us again soon.